which also comes with, for example, with the surface of the milling beads that I use. They have very often also a rough surface. And this also leads, this principle of using friction to grind something leads also to a certain usability of this mill. And this is something that I also wanted you to take from this lecture. For example, when you think about the ball mill, then you will automatically understand that it makes sense that very often you also get impurities in your powder, because when you mill something by friction, you have surfaces involved, you have also, it is automatically also clear that you have heat generation in this mill, because of this basic principle applied in the grinding process. You also have a certain amount of impact, but the focus of this mill is really that you have attrition, that you have in the end a lot of friction in this mill, which leads to also a grinding. And this is something that we were looking at for different mill types. For example, here we have a lot of pressure going on these powder particles. Here pressure is the usual thing that makes this particular mill so effective for certain things. For example, think about when you have something like, for example, a dried fruit of some plants or something. In this case, you can also use the compaction mill, because you have to break the structure and afterwards it will be easier to grind. So this is something where you can use pressure very often. Otherwise, when you, let's say, take a shell of, or you take just, for example, you take walnuts with shell completely into a ball mill, and you can mill them for, let's say, 24 hours, you will not see much. And the reason is because you didn't really respect what kind of force was needed to break this structure, and later the milling will be easier. In this case, I would advise just take a cutting mill or something if you want to really keep the shell and if you do not want to discard it. So this is the point that is important, I think, when we are talking about milling. You have one principle which is somehow embedded in this particular milling type. Cutting mill, for example, ball mill, for example. The jet mill, which is mostly working also on the base of attrition and impact. But it also has a certain principle here. It only uses the particles to grind themselves. There's never really impact on the surface of the mill, which means also you have much lower input or influx of impurities here. This is a thing that I mostly wanted you also to see when going through the different mill types. And here we, for example, also looked at compressibility percentage and flowability percentage, because it also, the particle size has a certain impact, and milling processes also generate that. And when I talk here about compressibility percentage, it means the compressibility index. Now you know that, for example, the effect of size is very huge, very often on the compressibility of the powder. That usually when you have a larger powder, you may see, for example, also a higher degree of compressibility here. But in this case, it is not a good thing, because it refers to the USP characterization of compressibility index. I think in this lecture we just quickly touched it. Later in the dissolution lecture, I really was going deeper into also the biopharmaceutical part. Just this is just the reference that you that this refers to is mostly noise Whitney. Surface area corresponds to dissolution rate, and dissolution rate in the physiological context very often also refers to better bioavailability, because we have a certain time window in the GI tract. This was also a part of the milling lecture. When we are talking about blending, I think some principles embedded in this lecture was mostly first to understand that there are different ways to achieve a good blend, and that we also try to get an ideal situation. The ideal situation usually happens when this ideal situation is also the energetically best configuration. For example, a drug solution. When you just put it into a solvent, then we are not talking about a powder, but this means the solvent molecules just group around the drug molecule because it's the energetically best configuration. In a powder, this almost never happens because usually the powder particles are always trying to, kind of form, or you will never really have an ideal mixture because it's mostly random distribution and a lot of forces involved. What you should also understand is what can lead to particle segregation in this case. And here we discuss two different effects which are particularly important. Brazil-nut effect. So the big particles cannot really penetrate deeper into the powder bed, and this is why they just end up at the top. But at the same time, we also have the effect of gravity, which means in the end also the big particles have, or there's a there's a tendency to sediment in the powder, and this also can lead to particle segregation. Usually, the Brazil-nut effect always has the overweight in these two, except you have the same particle sizes. Then the effect of the density of, let's say you have different particle densities, but the same particle size, then you have sedimentation. If you have very huge particles, usually Brazil-nut effect is the one which is usually the charge, which has just the higher impact on powder mixture. We talked also about the principle of the geometric dilution, which is just a way of also, I think what I said during the lecture is imagine a powder to be a dispersed system of a solid in a gas. And if you understand that, then you also understand that the energy input that I give on a certain amount of powder also matters, because I more or less increase the dispersing forces in this powder. This is what blending usually does. It just puts mechanical energy into a powder to achieve a dispersion. And if you understood that, then you also understand that later the amount of powder on which you put your mechanical energy matters. And this is the basics for geometric dilution. You just put more energy on a smaller amount of powder, and later you mix it again by increasing the powder volume later, or by including more powder. So you just make a mixture in several different steps. First you take the drug, then a bit of excipient, make a pre-mixture, and from that one you just slowly dilute it to the final amount of mixture that you want to have, the final ratio of excipient and drug. And this is something that you just do to also increase the energy that you put into your powder, into your dispersion. That's mostly the idea behind it. Is there any question on that? I think blending, I got no question via email on blending. I guess most people have a very good grasp of it. I think the only thing that some people don't know is that it's called the Brazil-nut effect, but usually you always eat the Brazil nuts first. So. When I talk about blending machines, then I very often also talk about the practical impact on your product. And this is also why I said, if you remember after this lecture a blender, then please remember the drum or barrel root blender, which is using mostly barrels to achieve a mixture. So this one here is just rotating, and you have a barrel here, and the reason is first you have an exchangeable blending chamber, so you do not really have any risk of contamination, and you do not have to clean much. And the second thing, most things that come into a production site are in barrels, so why not just use that to mix things, to put them together. And finally, we also had the very nice tubular blender. At least the micro people tried really to move their hands that way, like a tubular system. But I think it was also quite understandable. Any questions on that? I didn't get one about this lecture. If you have one, just come after. We have been going through the slides. So this is also what I want you to understand about blending. Mostly what are the basic operations. So you should understand it's a dispersed system. when I do the blending, then it is also, let's say, the principle is to give mechanic energy into a system to create this dispersion, and by that I also overcome all the other effects, like for example gravitational force. By just putting energy into that, I more or less have to work against the usual forces that act or that are affecting the powder. We particularly talked also about the effect of overmixing. This is something that is also sometimes not really, let's say, not really recognized. With common sense, you usually don't end up there that longer mixing times can sometimes also end up with a better, with a worse outcome in terms of the powder homogeneity. So the idea is that sometimes when you have an effect which occurs, for example, I think I talked during the lecture about magnesium stearate. The effect that you have there is mostly that the single particles that you have, in the beginning you have magnesium stearate there also in particle form, and over time, when you mix too long, these very often break and then coat the bigger particles, which means the adhesion forces between other particles that you usually would also like to have very low. These are growing. So the bigger particles are just pinned together by broken magnesium stearate particles. This happens because of two things. First, magnesium stearate has also, it's just not a very stable material. It can also break, but it's also quite sticky. And this means that over time you're just generating agglomerates, and this means when you mix too long, the mixture gets less homogeneous. This can happen. And this is an effect that is usually not really explained by common sense. This is why I also mention it. So, I think we also have pre-formulation. And I think here it is mostly about the physical-chemical characteristics of the drugs that you have. So what you want to know, I think we talked about pre-formulation as something that is usually done during the early discovery also, and that you have very small drug amounts that you can usually use in the pre-formulation. At the same time, you want to know many properties that matter later in terms of the biopharmaceutical behavior. And this means that you have to find out as much as possible with very limited drug amount. And there, I think, we talked also about these properties mostly. We talked about the pKa value, which is very obvious also from the chemistry. I think this is very clear, the log P value. Here we said it also matters if you have the ionized form of your drug, what forms matter. So this is also why you not always measure log P, which is only octanol and water, but also with certain buffer systems. We talked about different salt forms and that they can dissolve more quickly or let's say more quickly or more slowly depending on the salt form that you have. And we also talked about the polymorphic forms that are out there. And there I think I gave you the example of the HIV medication, ritonavir, which was really a case where a more stable polymorph was formed in the market, and this also led to completely different results in terms of also very basic properties. So when we are talking about crystal structure, and this is later, or this is the thing we are talking about in polymorphism. Crystal structure is really the structure which holds together single molecules, and this means it is so basic, so fundamental, that it also affects a lot of properties, like for example the melting point, like for example the drug solubility, and these are all the properties that you change by just having different polymorphs. Do you have any questions on these parameters so far? I also want to give you a bit of time to come down and ask me some more questions because this is more a tutorial, but I didn't know how to. Yeah, we don't do that in Germany very often. However, I will learn. So okay. So these are the things if you have a particular question, I think for the salt screening, what I showed is, for example, here for itraconazole, which is a polysoluble compound. I just showed different kinds of salts, and very often these salts, for example, bromeliate, is sometimes also used for getting better solubility. I think we talked a long time about solubility, but I didn't really get a question about that, so I kind of guess that this was also very clear. Just to ask you also, did you have already lectures on log P value? And I think pKa you definitely have lectures on. But log P or so, did you have anything on that so far? Okay. That explains it also, because when I get questions via emails, usually I would say the favorites were laser diffraction and trigonometry. But these are also, yeah, let's say, tough if you're not really in the matter. But for this one, I didn't really get any questions. Okay. Finally, coming to the solution testing. Okay. The solution testing is something, I also have a tutorial slide on that. When we are talking about the solution testing, it very, very often makes sense also to look at the same things, because it's an analytical assay proposed by the United States Pharmacopoeia. I think during the whole lecture, I at least had 4 or 5 quotations of the US FDA, which means regulatory context really matters in this case. For example, what I would also focus on, what are the requirements I can face, for example, what is, when I have a 1-point specification, what does the FDA demands? When I have a 3-point specification, what does the US FDA demands? These are things that are really essential, because they also pretty much depend on what product category you are in, and it is a regulatory requirement. And in the end, when you are really working somewhere in a formulation department, pharmacists are the quality guys. You are the guy who really later has to say, does that comply with the current regulations? Is that really what we want to develop? Is this really what, in terms of physiology and the regulatory background, makes sense? This is how you usually understand that. And this is the reason why, in this case, on the one side, the physiological background plays into that, because it is about safety. Why do we measure particular properties? Why are we interested in, for example, permeability? Why are we interested in dissolution behavior? And on the other side, we also have the quality aspect, because FDA requires us for particular dosage forms, one particular specification, for example, 1-point specification, release of 80% in 30 minutes. This is a specification set by the regulator, and this is why it matters so much, because everyone has to really comply with it. So I think I made quite clear where that all comes from. So I will not repeat the whole thing. Just a few comments here. So, first of all, the physiology is something that is very, very well understood for the oral route of administration, and this is also what can be really expected also from you to know, so that you understand, for example, it is later not really my release in the esophagus region, and this is why I don't test usually at a pH of 7.4 my drug formulation, a tablet, because in the esophagus I may have pH 7.4, but in the upper intestine, which is the more relevant one, I have a pH of 6.8. So this is also what I usually test when I want to know something about the dissolution and by that also about the bioavailability of the drug. You also have to kind of understand what are, let's say, the physiology behind it mostly. And this also means to understand a bit what makes the upper intestine so unique, what makes the small intestine so unique. And this, I think I made very clear, is mostly that it provides a very, very good site for diffusion. So it really increases diffusion, for example, by injecting emulsifying agents into this region and by increasing surface area. Permeability is something that we can estimate, and I think Lipinski's rules of 5 is something that you should have heard of. So you will, let's say, enter a formulation department somewhere if you, or let's say you can enter, but in the end it's really something that you should have heard of. So, and usually it is, or it provides an estimate of the permeability of a compound. And permeability matters because the ultimate barrier that you have to pass is a membrane. So finally, you want to know what drug properties are somehow related to that. That's important. This is a bit the historic background. I think this is quite clear. I made very clear what are the differences in dissolution testing. And this is also something where the regulatory background again matters. Regulatory background means in terms of USP, United States Pharmacopoeia, we are talking about quality control. In this case, you want to show that your product has a certain quality. This is where you use dissolution testing there. We have a second application, which is the one in formulation development. There you also want to see what happens under physiological conditions. And we have a third application, which is mostly the one for biowaiver applications, where you have a particular form of a drug that can be tested in vitro, and by that replace a clinical trial. And this is something that I also want to highlight a bit. Okay, I think the apparatuses are quite clear. They are not really complicated, are they? Just not if they are not complicated. Okay, no one else, but however, I guess so. I think a rotating basket is difficult to is not difficult to understand. So. Okay. Advertising videos, absorption. Oh yeah, sync conditions. This is something that I also took a bit of time to explain. So sync conditions is also just to mention it, if someone didn't understand. So sync conditions are conditions which make sure that you test in the in vitro test under conditions where you have a higher solubility or a higher maximum solubility, and this compensates for the absorption that usually happens in the GI tract. Now, the biowaiver, finally. So, the biowaiver is a very, very unique thing because it is I would say, the basis for our whole generics market. That we have generics is mostly because you can cheaply produce them. It also means that you don't really have to, pay license fees or something, but in the end you can save a lot of money by just taking reference to some other product in terms of safety. And this is why this is really an important thing, because in the end when you have something which has so much impact on the market, you want to know what it is. And this also means that biowaivers matter, but it also is important to understand the limitations. Because otherwise, every single company on the market would say, oh, I have something, it's a biowaiver, please approve. So in the end, this here is the definition of bioequivalence, and these are the requirements. And it's very, very clear why you have these requirements. For example, highly soluble compound. You do not want to have the solubility something to do with the general absorption because you show later comparability in the dissolution test, which also means that you show more or less the equivalence of two products. The tableting process should be the only thing that stands between this drug or which affects this drug being somehow absorbed, and this you prove. The dissolution test is something that shows the very effect of the formulation on the availability of the drug in a certain area. In this case, the GI tract. So, if the tablet releases the drug quickly, it should dissolve quickly, and it should go into the absorption phase very quickly. So, this is why we only apply bio-waivers for highly soluble compounds. We only do that for immediate release formulations. This has a very, very clear background because sustained release formulations generally also have a certain rate which matters over time, and there the rate is something that is also highly variable and also pretty much dependent on the manufacturing process we have. So here we do not usually apply a bio-waiver because bio-waiver says you only test the solution because it's the only property that matters. A wide therapeutic range. When you are in the regulatory authority, you are operator number 500 who's looking to dossier submissions from companies. The thing you are usually most worried about is safety. When, because of your decision to approve that product, someone dies, you hate it. I would too. And this is also why only things or only drugs with the wide therapeutic range are used in bio-waiver applications. Because here you say, okay, even if we had a slight difference in the release rate, maybe we have, let's say, 10% difference or so, it doesn't matter, because the patient will not die from these 10% or 10 less percent. And identical compound and similar administration route, I think that's quite clear. You cannot do that for two different compounds, and you can also not change the administration route because this just changes the ground rules. Okay. I guess this is what I had for you for this tutorial, because the other things are also very important, but I kind of got from the questions also that this was the things that mattered the most. If you have more questions, just come, and I'm sitting here now. So, then just ask questions.